So good morning, everyone, and welcome to day 2 of our innovation workshops, part of the On Helix 2022 conference. Thank you very much for joining us this morning. So the first session for today is going to be a member showcase. So we've got 4 One Nucleus members who will be presenting to us about their companies and the role they play in a sort of enabling translation, which of course is the theme of our On Helix conference. We've got a breadth of companies with this morning, which really helps to highlight the wide range of stakeholders that make up the One Nucleus innovation ecosystem. So just a couple of housekeeping points before we kick off. If you are not presenting or speaking, if you kindly keep yourself muted and cameras off, that would be really appreciated. How I thought we'd run it is we'll go through all of the presentations first and then do a Q&A session at the end. Please don't hesitate to drop your questions into the chat box throughout the session, and I will very happily pick those up at the end and pass those to the respective presenters. And as you will have heard, we are recording the session and it will be featured as part of the on-demand content for the conference, and you can access that via the conference app. So because we're running a bit of a tight schedule today, I'm going to jump into introducing our first speaker. So I'm delighted we have Kirsten Mclaughlin, who is Product Manager for Cambridge Crystallographic Data Center. So Kirsten, I'm going to hand the floor to you to share your slide deck. Thank you very much.
Okay. Can everybody see my slides?
That looks great. Just
Fantastic.
need to put them in full view. Thank you.
Yeah, of course. Okay, hello. As you just heard, I'm Dr. Kirsten Mclaughlin, and I work with the Cambridge Crystallographic Data Center. Today, I'm going to be telling everyone about the Csd Landscape Generator, which is part of our CSD theory suite. So I'm going to start off with something that is familiar to everyone, this sort of benchside chemist concept of molecule to medicine. So we start with our molecule, we transform that into a crystal structure, move that into microscopic crystals and then a powder, and that powder gets compressed into a tablet. The issue we're going to be looking at today is polymorphism. So this idea that we have our crystal structure, we've solidified our molecule into a crystal structure, but there could be other metastable structures available. And we don't know that we have the lowest energy structure. We could in fact have a metastable structure. Now this is a huge problem because, for example, in 1996, there was a new drug for HIV released, ritonavir. At the time, there were only 7 other drugs available for HIV, so this was important to the market and to the people who were taking it. When it released, they were using what they called later Form 1. In 1998, a new more stable polymorph Form 2 appeared. So this meant that it actually stopped working for the people who were taking it. It had lower solubility and lower bioavailability. They discovered that Form 2 was far more stable and that basically the presence of any Form 2 could trigger Form 1. And Form 2 was in their production lines, which meant that basically all the drug that was available had to be withdrawn. They had to shut down the production lines and they had to stop using this life-saving drug. They did eventually manage to reformulate. They replaced these capsules with a refrigerated gel cap in Form 1, but by then they had lost $250 million and people couldn't access this really important drug for a long period of time. That was in the 90s. It happened again in the noughties with rickettine for Parkinson's disease. And even as recently as 2018, there was a study by Newman and Van der Streek that estimates that between 15 and 45% of all small molecule drugs are one of these metastable polymorphs, and there's a more stable polymorph available. So as you can imagine, pharmaceutical companies are not sitting back and just waiting for this to happen. they address this with crystal structure prediction, CSP. So this is where we take the 2D molecule that we're considering, and we start looking at all the different coformers of 3D molecules that it could come up in. We take these 3D molecules and we put them into all the different possible crystal structures. Once we have all the different possible crystal structures with all the different 3D molecules, we sort of do a free energy calculation and looking at, and generally we look at it in terms of free energy versus density. And we have down here the sort of area where the lowest free energy, highest density polymorphs are the most stable. And you will hopefully get a clear one that is the lowest energy, highest density, best crystal structure. So traditionally, this is done through first principles. Now, as you can imagine, all the different conformations, all the different crystal structures, based on first principles, this is really computationally expensive. We're talking 10 hours to several months on pretty serious hardware. And again, basing it on first principles, it requires a lot of expertise, because if you are running this model for several months, you want it right in the first place. So we're talking about a really high cost. So one landscape can cost up to $100,000 to run. And because they're so high cost, they're performed later in the pipeline when the company is willing to invest large amounts of money into this medicine or this potential medicine. At the CCDC, we've produced this alternative method, so a Csd Landscape Generator. And this is taking an informatics approach rather than first principles approach. We're using the Csd Cambridge Structural Database as sort of basis for these. And again, we're starting with a 2D molecule, but the 3D molecules are generated using bond angle and torsion information that we already have in the CSD. And similarly, the crystal structures are based on the experimentally confirmed structures we already have. Finally, the landscape itself, the free energy, is calculated based on sublimation energies, again, that we already have in the CSD. So this CSD, Cambridge Structural Database, just for some background on it, it is sort of the core that CCDC is based around. We have over 1.1 million experimentally verified structures. We get these from publications, but also early publications. We have our own set of CSD communications for places where people want to publish a structure, but not an entire paper around it. We also go through patents, university repositories, and theses for them. We have a team of experts who go through all of these, enrich them, verify them, check them through. Yes, this has been going on for 56 years now, so it is a huge repository of all this information. Obviously, CSD is at the core of everything we do, and it is sort of what we focused on for this landscape generation. But we also have it set up so that if you have proprietary crystal structures within your own database, you can use that as well for this landscape generation. So if we just go back to that computational approach and the issues with it, we are no longer basing it on first principles, which means we no longer have a computationally expensive system. So as opposed to 10 hours, my computer can run a landscape in about 20 minutes. And it no longer requires all this expertise because that expertise is embedded in the database itself. And that means it's no longer high cost, and you can do it earlier in the pipeline rather than waiting until you're fairly committed to the molecule. And that sort of earlier in the pipeline concept is something that we really push forward with CCDC, in that we want to approach these things as digital first rather than starting at the molecule. So the idea that you can generate all of your conformers, you can look at your crystal structures, but you can also even start looking at, okay, how well is this going to crystallize? What are the form of the crystals going to be like? What are the surface properties of the crystals going to be like? How well is it going to compress into a tablet? And you can sort of identify where the issues will be for the bench-side chemists, and if you want to go forward with the chemistry based on the issues you're going to face later. Okay, I had a backup tutorial here in case everything went wrong, but it's okay. So I'm just going to do a quick demo. Now, as you can imagine, 20 minutes, well, really fast and really great, doesn't work well in a 15-minute demo. So I have run this beforehand. So this is, let me make sure I can pronounce it correctly, sulfathiazole. And I just earlier today imported it using a SMILE string. So you can bring molecules in, SMILEs, INCHI, you can sketch it out, or you can load it using a zip file. I brought it in and it's underneath there. Sorry, the zoom bar is there, CST theory, landscape generator. And there we have it. As you can see, generate. It is not an onerous, heavyweight dialogue. It's just a couple clicks. And then, like I said, 20 minutes to generate it. So if we look at sulfathiazole, I imported this earlier. It shows a conformation to present in, but it is going to calculate all the other potential ones. And then, let's put that down there. You can see that we have all these different predicted conformations. And we can see that, for example, each one of these is a sort of it does have all the sort of relevant data associated with it. So we can start looking at, okay, how would this pack together? What kind of H-bond network does it have? And start going into, like I said, surface analysis and looking at the different sort of particle shapes that would be associated with this. Now, I'm sure nobody wants to go through 100 different predicted structures. So there is, of course, as well, a data analysis tool looking at the different structures, what the density is, and those sort of absolute score, those enthalpy estimations. We can go in and we can do, for example, a scatter plot looking at density versus that absolute score. And there we go. And you can start seeing here, we have our low energy section, low energy, high density. And if we click on these guys, you can see that it gets highlighted here and here. And we can start actually taking a look directly at these structures and how they behave. So that's a bit about the tool. And of course, you're welcome to ask for a demo and see it in more detail. We didn't do this on our own. We did this as part of a consortium. Oh no, start at the beginning again, sorry. We did this as part of a consortium. The Crystal Structure Prediction Consortium with industrial partners, academic partners, professional bodies, and other software providers. So just some of the different participants that you can take a look at. Yes, and thank you very much for hearing about our latest release.
Excellent. Thank you so much, Kirsten, for that exciting update into the advances in CSP. So there are a couple of questions, but if it's okay, I'd like to move on to the next presentation and then I'll get to the questions at the end.
No
so with that, I'd like to invite Gavin Yates, Program Director at Catalan, to start sharing his screen. And yes, over to you, Gavin. Thank you.
Thank you. Just split my screen. Okay. Okay. Good morning, everyone. My name is Gavin Yates from Catalan, and I just want to talk to you about the clear advantages to using an integrated team with development and clinical teams working together to achieve success, and the considerations that we should be thinking about when we are talking about our early phase planning in our clinical trial through to later phase. So I just wanted to start with the question, why is speed to the clinic so important? So an integrated approach using a development and clinical team supports all different sizes of pharma companies from small to large. But what we're seeing within Catalan is we're seeing a number of emerging small biopharmas looking at new first-line therapies. And within this area, budgets are ever tightening. Companies will have the direct milestones to hit for further investment to move to the next phase of their project. So hitting these milestones quicker is so important. And the faster we do this, the quicker we get additional funding that can be obtained for the remainder of the project and to move to the next stages. Now, emerging biopharma companies also can have internal constraints. There are within the teams, team members will be wearing multiple hats to support the drug development cycle, and that can dilute certain areas. So when the expertise are diluted, decisions on the trial setup can be made that may not be the most efficient for conducting the trial. There are a number of new technologies and innovations out there that can support patient recruitment, retention of patients on the trials, and actually engaging in the right subject matter experts earlier is so important because that will help drive success of that trial. And as we're seeing across pharma, trials are changing. They're becoming more complex, targeting niche areas, as I mentioned earlier. And it's causing us to have to think how we set up those trials, more complex trials differently, new ways of creating efficiencies, as I mentioned, accelerating timelines to reach the patient, but also making sure that we reduce wastage of the material. In addition to that, we're all seeing at the minute, as well as the increasing number of trials, the supply chain challenges across the globe, increasing competition for critical materials. So understanding the scope and the end deliverables is more important than ever to achieve success. So no matter what size of the company, for the clinical sponsor, there can be a lot of competition for recruitment of patients, for obtaining critical materials, and to help ensure that that finished kit can be delivered to the patient to support patient enrollment as quickly as possible through the clinical phases and actually be the first to market. So as I mentioned, speed is such an important factor, and there's two commonly applied approaches sponsors implement to get the speed they need. So the first one is timeline compression, so expediting certain steps. Now, simply doing this as fast as possible, but keeping the traditional more sequence of moving through the project one step at a time. The second approach is creating speed through parallel processing, and that is where various steps are done at the same time wherever possible to reduce waste, to reduce the overall timeline of the program. And when we try to accelerate that first path to the clinic just by compressing and expediting the timeline, most of the buffer in that plan is immediately eliminated. So because of that linear approach of that compressed timeline, waiting for one activity to finish before starting another one, if something does not go entirely to plan, and we all know within drug development and manufacturing and clinical supply, there will be certain times where things don't go to plan. And because of this, this means that there will be a domino effect and ultimately delaying the delivery to the clinic. So on the screen now is a typical linear timeline where you look at, development of your drug, manufacture of your early phase product. So working on one part of the project at a time. So you start with development. Once that's completed, you go to your drug substance process development, manufacturing, look at the manufacturing approach and understand where the quality attributes, inputs and outputs are identified. And when these activities are completed, the next phase is then to perform the scale-up. Apologies. Manufacturing a batch usually in the way of engineering is a final step before moving into the to GMP product manufacturing of patient products. So as you can see on this slide, these activities are commonly performed in a siloed fashion. And in the past, what we're seeing is that there's many examples where you have your GMP manufacturing step or setup complete, and that's when the clinical discussions begin to start looking at your packaging design, sourcing of comparator, how the QP strategy will work, and how your storage and distribution supply plan will be put in place. And that is becoming too late in the process. So what we wanted to show you is how we bring the right subject matter experts from clinical into the development discussions earlier at the right points, so we can help ensure that there's a streamlined approach to simplify development, accelerate timelines, and reduce risks of your clinical programs. So here we see on this slide the same activities, but as you can see, we're now overlapping all of the clinical supply activities with those of manufacturing. So getting a major head start by initiating packaging design while still in the process development stage. So it's in this phase of development that it's highly likely we understand the drug characteristics. You know, are we looking at, does the drug have certain temperature conditions, light sensitivities, humidity sensitivities? You know, and being armed with this information, the clinical supply team can take this and start activities and taking certain activities off the critical path. So if you're looking at sort of the long lead times for designing and ordering of booklets, for example, for your global trial, or ordering packaging components with long lead times, with the ever-extending lead times and supply changes we're seeing across the globe, that can all be discussed earlier and taken off the critical path. So as you move through the entire life cycle of the program, you'll see that the activities such as like comparator sourcing, storage and distribution setup can all happen more in parallel, so that once material is manufactured and released and delivered to your clinical site for packaging the finished kit, it can move straight into the production, so accelerating your timelines. And the benefits, other benefits you can see is that the teams from development, manufacturing, and clinical are all working as one project team. So you're reducing handoffs, integrating their activities with supporting strong communication along the way. So risks are identified and mitigation plans are put in place. And this approach also creates buffer to the schedule because the teams are effectively planning and can avoid delays that may arise otherwise. So the final result is actually a faster delivery to the clinic and to the patient. So as I mentioned, engaging earlier is so critical to understanding your success to the trial. You know, you want to get your drug manufactured, but you know, you will need to get that drug to the patient. So let's talk about what questions we should be asking regarding packaging and storage and distribution earlier on. And by getting, as I mentioned, the clinical team in earlier, during the dose form development selection stage, we can design the packaging to support characteristics of the drug. We need to consider as an option to protect the drug, and the other is ease of use for the patient, you know, dependent on the indication of the trial. Now, handling examples include, for example, handling of the drug and how it will be packaged using like cold room facilities or potent suites, what the time out of excursion for each drug would be, and how this will be controlled and monitored. If there's any specialized labels with advanced adhesive on your vial and ampules for frozen conditions. There's creative packaging, you know, for fragile vials for, you know, global distribution studies. I've seen a recent example where a patient kit was hundreds of thousands of pounds just for one kit to the patient. So the team engaged earlier to design a kit that created this extra support, but allowed actually ease of opening of the kit at the clinical site, and extra layers in place in case there was any leakage due to broken vials during the shipment. You know, think about the resupply. So once the first manufacture has taken place and we've started enrolling and dosing patients in your trial, how do we plan then for the next manufacture to make sure that the patients continue supplied with drug? Is there enough storage at various locations? If you're, again, performing a global trial, you know, at the start, you may only need a small amount of storage for your early phase. As you move to the later phases, that is likely to increase. is the setup or is the capacity available to support that as you move through your later phase trials? And expiry updating if there is limited stability of your drug. How do you plan that in to make sure that you can expiry update as the stability time point results come through? We also need to think about distribution, and I see this being overlooked a number of times as well. You've got your drug manufactured; it's now actually packaged into the final kit, and then the companies are looking at how then they distribute to the patients. Now we need to think about this, preferably months in advance to understand if the drug has a certain temperature. Do you want to use a specialized shipper system or specialized couriers that can be distributed to remote areas? If there is a limited drug supply, there is specialty distribution methods that we can use to reduce waste. We can actually ship directly to the patient in some regions of the globe as well. So straight from the clinical packaging site to the patient to support patient retention in the trial. Once all the patients have taken their drug, how will the returns be processed and how will you destroy the remaining stock? And what about import of record requirements? So when we're shipping from one country to other regions, how would we support the import of records across that? So by discussing these points earlier in the supply chain, we can get set up and ready to go. So as soon as the kits are released, it can be shipped to the patient. So we've talked through the concept to getting the material to the clinic quicker, but how can an integrated approach streamline and accelerate to get the drug ready for the clinic? We have within Catalan a team of clinical supply managers who help coordinate communication between the sponsor, the IRT, manufacturing, clinical project teams, development teams, so that important factors related to the whole project can be evaluated up front. So drug characteristics, packaging, and distribution strategies, and very importantly, material expiry at the patient recruitment plan is understood. So this sets up the project for success and ensures continued supply to the patients. And I want to just talk around an example now that I've experienced where the clinical team and including the clinical supply manager was not engaged early and there was challenges in the study. So once, again, the drug had been developed and was going through the manufacture, the client engaged with the clinical team. And it was 4 months before they were planning to dose their first patient. Now, at this point, there was no discussions on packaging materials, how the project was set up, and supply chain. Now, this should have been discussed months earlier, as shown in my slides earlier in the presentation. Now, when we did engage with the clinical supply team, it was actually, and we reviewed the protocol, it was uncovered that the initial manufactured quantity would not meet the protocol demands for the trial. So immediately there was an urgent rush to perform another manufacturing run to gain more bulk drug for this project. In addition, there was limited stability. So the expiry of the drug would not last the length of the trial. Therefore, expiry updating would be needed at the appropriate site locations. But that all wasn't included in the submission to the relevant countries. So an amendment needed to be updated. The submission needed to be updated with an amendment for the teams, for the sites to be able to perform expiry updating of that drug, again, as the stability time points came through. And there was no discussion on import responsibilities to the relevant countries where this material is being shipped to. And as it was an emerging biopharma, they did not have the right entities in the countries for the material to be shipped. So we needed to work out a way to supply that material to the relevant countries to dose the patients. So this impacted the start of the trial and shows the importance of using that integrated team to support the full drug development cycle. And by engaging earlier, then these teams could have discussed all these actions and put plans in place to prevent any delays. And we don't want to spend years of effort developing and manufacturing the drug only to see in the final stages getting material to the patient is a hurdle. Now, one of the last slides and points I want to make is is also to plan ahead and plan for success as you're moving from early to late phase. So early phase, you may be looking, where you're setting up your trial, one site, maybe 2 sites in one region, and there's plenty of companies out there who can support manufacturing and packaging for that small phase, low quantity, simple distribution activities, but you want to plan for success. So think about past the initial phase 1 and focus on the next phase. So by working with a company with a global network, it provides sponsors with options and flexibility as the drug moves from early to later phase, allowing access to different regions. So supporting manufacturing clinical activities with the relevant regulatory guidelines of those regions. So you can see on the slide, there are, as you move from the phase 1 to the phase 2s, you may look at doing US and Europe, or Europe and Asia. And if you have a selected partner for your phase 1 that has the same processes across all sites over all these different regions, that phase 1 model can be replicated at other sites in other regions. So you've got a seamless transition as you move from phase 1 to phase 2 and beyond. And in addition, by using a vendor with the global network, you can add sites, all the sites into your submission, meaning that projects can be moved to other sites if required. And what we've seen, over the last years with the pandemic, it's so important to have contingency planning in place. So imagine if a site is needed to be, shut down in certain regions for restrictions, as we've seen in the last few years, and that project needs to be moved, having a global partner with a global network that can facilitate the move without, which is invaluable, and there's no need to set up new agreements, project teams, processes in place. It can have that seamless transition to another site in an unlikely event like we've seen in the last few years. Okay, to summarize, I really hope I've shown how important it is to look holistically at the project and providing that material to the clinic and to the patient. Just a few takeaways from me. The first is how important it is to bring your subject matter experts in all throughout the process. And when you have a team looking at it from different perspectives and with different backgrounds, they can come up with unique solutions to problems. I worked with Catalan now for a number of years, and there can be a solution to support the trial, and by getting involved earlier, there is more time to come up with that solution. Teams need to communicate and need to collaborate, and having that integrated group together and have them interpersonal relationships can really help facilitate that. Second is streamlining. As I mentioned, if we don't get to the clinic, we don't get it to the market, and we know that sometimes in early phase development that we're working on tight timelines to meet those milestones to get to the clinic. So streamline, eliminate as many handoffs as possible before you introduce risk, because that's introducing risk, sorry. And the third point is to look how new innovations within the industry will help deliver success for your project. And lastly, consider how today's plan will meet the near goals, but how that plan can take you to tomorrow as well. So build your team that will have these personal relationships. Never underestimate personal relationships within the companies. Working as a partnership, it's so very important to be part of that team, and for all companies in that project to be invested in its success. And at the end of the day, it's all about getting that drug to the patient. So that's what we're here to do. So I'd like to thank you very much for your time, and I will turn it back over to the host.
Thank you very much indeed, Gavin, for that very comprehensive overview of sort of streamlining and integrating the clinical manufacturer. There's a lot there to consider and think about. There's a question or two that's gone into the chat box. Again, if we have time at the end, I'll pick those up. If not, I'm very happy to connect those with the questions to the host. So I'm very swiftly going to hand to Yelena now from PharmEnable, Chief Business Officer, to give us an overview of PharmEnable's AI approach to drug discovery. Over to you, Yelena. Thank you.
Lovely. Thank you very much, Alicia. And yeah, delighted to be here today to tell you a bit more about PharmEnable and our approach to drug discovery. So just to give you a little bit of context about the company before I kind of delve into our technology. So we're a relatively early stage company. We're focused on using AI to improve drug discovery, particularly discovering small molecule drugs for otherwise undruggable targets. We have our own early stage drug discovery pipeline, and we develop assets either as wholly owned or through co-discovery projects and collaborations. Our wholly owned pipeline is focusing on challenging drug targets in the oncology area. And then our co-discovery partnership programs and collaborations focus on challenging targets across different disease areas that could benefit from novel chemistry. We do a lot of work in neurology, for example, because our system is well suited to predicting molecules that work well to cross the blood-brain barrier and have other favorable properties. So Alicia mentioned at the beginning this idea of the kind of Cambridge innovation ecosystem, and we're very much, as a company, we're very much a product of that innovation ecosystem. So all the founders met in Cambridge. We were originally a spin out from the Department of Chemistry based on medicinal chemistry insights on how to build up complexity in small molecules in a small set of synthesis steps. So how do we get small molecules that are more complex and three-dimensional and more nature-like than what's traditionally been included in screening libraries. And we also have a very experienced leadership and board. So for example, our chair, Dr. Jane Dancer, is also Cambridge-based, and she has a lot of experience building and exiting particularly platform companies. And I also work closely with Gaynor Friars, who used to be the VP of Business Development at AstraZeneca. And we've had fantastic support from a number of Cambridge investors as well. So we're very much kind of part of the innovation landscape here. So in terms of our mission, I expect that all of us here today will have had someone we care about affected by cancer or neurological disease, and also the associated treatments for them. So sometimes there either aren't effective treatments available, or sometimes the treatments themselves can be devastating and have a lot of side effects. So the mission of PharmEnable is all about finding better solutions for that. So how do we design better drugs with fewer side effects and with increased efficacy? And so if you imagine for a second the universe of all possible small molecules that could one day become drugs. So this universe is massive. It's estimated to be 10 to the 62. It's really vast. It's estimated that at the moment, only about 1% of it has been explored. If you're working on challenging targets, it might be that some of these kind of existing screening collections and approaches only target a part of the chemical universe that doesn't necessarily work that well for that target. It's a little bit about a matter of luck. So what we wanted to do is create a tool that really taps into the entirety of this possible chemical universe and is able to find, for any given target that we're presented with, is able to find the best part of that chemical universe for that target and generate those small molecules and use them for research. So what's really unique about the system and what's originally kind of based on research from the University of Cambridge is the method by which we assemble the molecules. We've got this collection called Chem Universe, which is a human-curated collection of medicinal chemistry knowledge, so of different strategies and sets of rules for building up complexity and three-dimensionality within molecules. So what's quite unique about our system is that rather than having a fixed virtual library, which a lot of places do, instead what we do is we use our collection of rules to generate dynamic libraries. So we'll identify a part of chemical space that looks promising for a given disease target, and then we'll populate that part of chemical space with molecules, even if not a single one of them is known right now. And we do that from kind of basic building blocks and our knowledge of chemical strategies and reactions. And this means that we're able to tap into parts of chemical space that are underexplored and more novel and complex. The reason that we're really focused on this is because we believe that complexity and three-dimensionality can have a really big impact on the efficacy of drugs. So it can give us some improved properties in terms of specificity, in terms of efficacy, having decreased toxicity because of decreased off-target effects, and really tapping into diversity as well. So how do we tap into different chemical structures, different areas of chemical space that can be used as this kind of treasure trove of potential novel therapeutics. So in terms of what our system looks like, so we start with a particular therapeutic problem, and from there, we identified a part of chemical space that's particularly promising for that specific disease target. We populate it with molecules. We then rapidly drill down we start from billions of molecules, but really drill down to a very small number of them. So typically we'd predict between 10 to 30 molecules. We then synthesize these, test them, and feed the experimental data back into the system for a few different rounds of optimization. So this looks like this. So first we start with a particular therapeutic problem. So we can work from structural data, we can work from ligand data. If there's no structural data available, we can work from homology models. Generally, we need some data around the target in order to be able to work on it and feed that into the AI platform. So the first thing that happens is that ChemSailor, our AI platform, is a kind of mathematical map of chemical space, and it finds a particularly promising target area. We then use our knowledge of chemical reactions to populate that very promising area with billions of molecules. And sometimes we generally focus on novelties, so molecules that aren't present in other commercial screening collections and that aren't patented yet. And finally, we have a kind of multi-parameter optimization algorithm that's able to go from these billions of molecules very rapidly to a very small number of molecules that works very well. And because it's a multi-parameter optimization algorithm, it means that we don't just focus on efficacy, we can focus on a range of properties. And this is also custom defined for any given project that we do. So it means that we're able to really customize the properties of the molecules that we're designing to a particular therapeutic problem as well as to the target. So just wanted to tell you a little bit more about our collaboration with Denali as well. Thank you very much to Nucleus and On Helix for listing us as one of the finalists in their By News Round Awards. So this is why I wanted to touch on this. So this is a strategic collaboration that we launched in June this year. And the aim is to accelerate drug discovery in neurodegenerative disease, which is an area of enormous clinical need that really needs new solutions and new drugs. And it's a joint research program against a number of different targets over the next 2 years. So we're really excited to be working with Denali for a number of reasons. We're both very much mission-driven companies, and I think that's something that we very much have in common. So everyone within both companies really cares about patient impact and ultimately getting new drugs out there to patients. So it's something that we're really aligned on. Denali brings really kind of significant in-house knowledge and proprietary data around targets in neurodegenerative disease. And on our end, we're really kind of chemistry specialists. So we develop challenging chemistry against well-characterized targets. So in our case, we bring our proprietary AI platform to enable the exploration of novel chemical space to really try and unlock these targets. And finally, so Denali is kind of highly experienced in successfully engineering delivery across the blood-brain barrier to drive a therapeutic effect in the brain. And something that's complementary on our end here is that our optimization tools help create molecules that are really designed with that in mind. So we look at molecule properties to make sure that they're well suited to both the target, being very specific, and also having the right types of properties in order to cross the blood-brain barrier and work well for neurological disease. We're a very collaborative company. We're always open to talking to people, and we're kind of generally open to talking about potential co-discovery partnerships and collaborations. So what we bring is an AI-enabled human-designed approach to drug discovery. So we do these kind of cycles of using AI to really boost what our chemists can do, but then also having the sort of human input and to add the kind of scientific rigor and selection to parts of the process as well. And we bring the ability to identify target-relevant chemical space that might not have been explored before. And as I said, we're very collaborative as a company, so we'd love to hear from people. And what we're particularly looking for is kind of companies with expertise in disease biology and particular targets, with complementary skills and knowledge, but where the target has been well characterized, but very difficult to drug. And our expertise is in finding small molecules that can unlock those targets. And thank you very much. So if anybody would like to get in touch, please do drop me an email. Always happy to have a chat. And thank you, Alicia, for hosting us as well.
Excellent. Thank you very much indeed, Yelena. Yelena has to drop off at 11, but if there are any questions, I think one went in, I will join the dots post-session. So moving on to our final presentation for this morning's session, it is Tim Holden, Commercial Director, and Mike Savile, Director of Industrialization at Cambridge Pharma. So over to you guys. You can start sharing your slides. Thank you.
Good morning. Okay, thanks, Alicia. Let's try and share these slides. Is everyone seeing that?
I can just see it said started screen sharing, but there's no slide shown at present.
It just completely froze. Is there anything come up now?
Not as yet, no.
It's just completely frozen.
I'm more than happy to share the slides if you need me to.
If you can do, yeah, because it's just completely froze this end and I can't actually do anything. Brilliant, thanks very much. Okay, so what we'd like to tell you about today is a new business that we're starting here in Cambridge. So Cambridge Pharma, Pharmaceutical Development Services. If you go to the next slide, please, Alicia. So this is part of the SMC group of companies, the medical group of companies. So it is associated with Oval Medical Technologies, who design and develop drug delivery systems, also here in Cambridge and just across the road from our new site. And we are associated with SMC Limited, manufacture and assembly of drug delivery devices and other medical devices in the US. And then Cambridge Pharma, the business we'll speak about today, which is pharmaceutical development services and fill finish capabilities here in Cambridge in the UK. Can we have the next slide, please? Okay, so the basics of our services here, small batch sterile filling. So we IMP license, we've got a flexible service using isolator-based filling suites. Including the release to any clinical trial sites and associated companies for your clinical trial. We also have the capability to do process development, stability testing, QC, chemical analysis here on site in Cambridge that allows us to support those sterile batches that are being manufactured. And the facility that's built here, we'll go into a bit of detail later on, it's a state-of-the-art, brand new GMP facility that has a energy efficiency system built into it to run the clean rooms, which helps reduce to your carbon net zero goals. Next slide, please. Okay, so pharmaceutical development services. So what we look to do is help you develop the fill finish process for your product. Many products going into fill finish and clinical trial supplies have degrees of development that have been performed up to that point. So we have the flexibility and the ability to help get that up to a GMP standard ready for sterile fill finish in the GMP facility. As mentioned, we also have supporting QC chemistry labs, and the ability to do stability testing. Also support this with QP release and engineering and validation support teams on site. We also have capabilities in development labs here on site to develop processes to do with, for instance, container closure or any physical device testing that needs to be carried out, associated also with any human factors work that needs to be supported that for a drug delivery system, for instance. Next slide, please. So starting off this process, we have a, say, dedicated on-site process development lab. So this is allowing to do any of those development activities outside of GMP to support a process readiness to go into a fill finish operation. That can be all the way from the actual fill finish process itself, the compounding process of the formulation to go into that primary container, any associated device that may go with that. And it's a lab that operates to GMP principles, and also 13485 for any devices that may be included in that. And it's a clean but non-controlled lab, so we have the ability to make clean batches under unidirectional airflow areas. And then it's supported by the Analytical Lab for testing any products within that. So a very flexible space to support early development. Next slide, please. Thank you. So across the road, we have the manufacturing facility. So this is a GMP licensed site for IMPs. It's a dedicated sterile manufacturing facility, so approximately 10,000 square feet. Supports 2 clean rooms that support isolator-based lines. And then the rooms are grade C with the grade A isolator lines inside them. Next slide, please. Okay, so one of the critical factors in transferring a new product into manufacture is the tech transfer process. You know, the experience of the team has based, you know, in previous lives of this is a critical phase to do that transfer. And we, you know, sort of understand that companies may be in different states of development when they come to transfer into Cambridge Pharma. So we have a structured tech transfer process that we walk through with all customers to transfer into that clinical trials manufacture state. And it allows us to de-risk that activity. So when you actually get to manufacturing that sterile batch, you will be more successful. Okay, next slide, please. So this is the clean room facility. Say it's a new build facility, state-of-the-art, two GMP filling rooms. As I say, they're grade C background with grade A isolators inside there. We support those sterile filling lines with a on-site autoclave to allow sterilized product that's going into those, be it for aseptic or terminal sterilization. We also have washers on site to support any of those materials that are going into that site, into those lines. Sorry. Next slide, please. Okay, so the clean rooms themselves, they are supported in terms of the HVAC system by a novel control system, say that is aiming to reduce the environmental impact and energy usage. So we partnered with a company called Eco2 to install this system. The intelligent clean room control system allows a dynamic approach to controlling the particular ISO standards within the room. And these happen throughout the production. So in times of downtime, when there's no activity in the room, then the whole system's ramped down to maintain those levels, because of course there is less generation of particulates in the room at this stage. So overall, it roughly reduces energy consumption by around 60 to 70%. Next slide, please. Okay, so 2 filling rooms, slightly different approach on each of the filling rooms. Filling room 1 is more dedicated to smaller batches. So it's a more flexible approach, running different modules that can be moved in and out of the isolator line to cater for different products, different container types, and different fill volumes. It's a slightly slower rate because it is a semi-manual process, but all of the critical activities are still automated by stations that you see on the screen there. So typically around 1,000 units per day, dependent on the process. Next slide, please. This second filling room is a more automated based line, so it's a nest-based filling line. This includes a hard-walled isolator, which has VHP decontamination also. It can cater for different primary containers, but is more aimed at sort of later stage when the volumes get larger. But it has additional facilities in there, for instance, like 100% IPC weight check of all product. Typically, you're going up to around 5,000 units per day for this particular line. Next slide, please. As mentioned before, we have an on-site analytical lab to support the activities within the fill finish batches. So this is a dedicated testing facility. We have HPLCs, particle size analyzers, all sorts of chemistry analysis equipment that you may need for that particular product. But also we can support any mechanical testing, for instance, if there's a device involved, for instance, a drug delivery device that requires some mechanical testing. We also have ICH stability chambers on site for any stability standards that need stability trials that need studies that need to be carried out. Okay, next slide, please. So we also have a packaging inspection area within the facility, so we can support the visual inspection of the containers that are filled during the batches. We have ability to develop and carry out integrity testing, so CCI testing of primary pack of various types. And if you have any specialist closure systems, we have the capability to develop those methods with you as well. And then we can also do secondary packing, cartoning with IFUs and packaging for transport, for instance. Next slide, please. So just a summary. So it's a fast and flexible service to help you meet your clinical trial deadlines. We very much work with customers from the state they're in to get up to a position where you can actually manufacture your clinical trial supplies. We look to have that flexibility to cater for any stage that you're at. As I say, it's a new facility. It's based around isolator technology, so it's the highest standards of sterility assurance. Yes, we look to build a partnership with your team to minimise that time to the clinic.
So come and see us. Make an appointment, come and see us and we'll show you around.
Okay, thank you.
Excellent, thank you very much indeed. There's a lot of emphasis on quality there, which is of course very important when you're looking to partner up with on your drug discovery programs. So thank you very much indeed, guys. I'm just going to stop sharing there. So we are up to time almost. However, there have been a couple of questions dropped into the chat box. So I'll try and pick up a couple and if we can get some quick answers. So Kirsten, if we still have you on the line, we've got a question here. The crystal structure is only one form of the molecule. Are there examples where it does not predict behavior in solution?
Yes, so fortunately I got this question early so I could message my boss to find out the answer. His answer is, it is very true that crystal structure is only one form of the molecule and that multiple conformations will likely exist and be explored in the solution phase. Whilst we can inform behavior in the solution phase, we don't currently actively model the solution phase using our products. The most likely conformation in the solid state is not necessarily the same most likely conformation in the solution or gas phases, although the range of options and the underlying energetics are likely to be broadly the same. I can send over his full response, which was several paragraphs long.
Excellent,
later.
Thank you. I will join the dots. There was some more, so I will make sure I follow up. Yeah. I'll just throw one over to Gavin because there was another one for Gavin. So whilst integrated development sounds good, is one of the challenges for companies to commit to the cost of such an approach?
Yeah, it was a really good question. My response back to that would be, if you're engaging with the right subject matter experts early enough, you can understand what the drop dead dates are to for example, start planning or ordering of certain steps that's on the critical path. So there is going to be a time where you will need to perform that step, whether it's ordering of components or setup of your supply chain. And by getting involved earlier, what you can do is plan for that stage. So you can say, you know, if you are looking to have this patient have their first dose in 6 months from now, you will need to order these components at least 8 months from now, or you'll need these to start thinking about your supply chain 4 months from this point. And the idea is you've got that in your plan, you know what you're looking towards, and when it gets to that point, there is a decision point that needs to be made, and you can look at a review with your development, manufacturing, clinical project teams, what the information that's available, and make that decision whether either to hit the button and start that planning or ordering at that certain step, or we need to look at potentially reviewing the first patient in date. So it's all about understanding information earlier on to plan, and just if there's certain steps on the critical path, we can even just look to order or pay for those just to make sure they're off the critical path, and the remaining, you know, parts of the project don't need to be you know, quoted for or discussed until they've hit their drop dead dates, if that makes sense.
Excellent. Thank you very much, Gavin. And over to, I think we've, Yelena's had to drop off the call. She's got a hard stop at 11, but just one for Mike and Tim. So as I sort of highlighted, you know, quality is a really important factor when you're looking to partner on your drug discovery development programs. If you could say the top way you demonstrate this, what would you say that is?
So I think it was sort of mentioned on the slide deck there. When you are doing the transfer in of a particular project, that is one of the highlighted areas of risk where you're quite a high probability to fail, and that's unless that process is very thorough. And that's why we've set up a structured approach to doing that, to de-risk any downstream activities. You know, once you get into validation of the particular process, that is a given that you're going to have sterility assurance of the highest level. It's the lead into that to make sure you've got a high quality process to transfer in, which is probably one of the number one we look at.
Excellent. Thank you very much. Thank you all for those succinct answers. So I'm going to close the session now and just say another thank you to Kirsten, Gavin, Yelena, Tim and Mike for their participation in the session today. We will be returning at 11:30 with another 4 presentations from our network and members. So I do hope you can join us then. Please use the conference app to connect with any of the speakers that you heard today if you'd like to follow up. And if you were attending in person on Thursday, I look forward to seeing you then. So thank you very much, everyone. Take care.
Thank you.